This is about interferons. Interferons are the proteinaceous antiviral substances. So these are the protein substances and they are antiviral in nature, and they are also species specific in nature. And these interferons are produced by mammalian cells. Not all the time interferons are produced by mammalian cells, but when our body cells are infected with viruses, then at that time interferons are produced. And interferons were first discovered by Alec Issa and Jean Linderman in the year 1957. And interferons are classified as alpha, beta, and gamma. And this classification is based on the structure of the interferons. What these interferons do in the body is they stimulate when our cells are infected with viruses. These interferons stimulate the cellular DNA, that is the DNA present in the cells. So the DNA of the cells are stimulated by the interferons so that the DNA, cellular DNA, can produce antiviral enzymes which stop viral replication. So whenever there is an infection by the viruses, interferons are produced, and these interferons are stimulating the DNA of the cells to synthesize antiviral enzymes. So these antiviral enzymes, which are synthesized by the stimulation of the interferons, they stop the viruses to get replicated so that the cells are protected. So this is the function of interferons, how these interferons stop viruses from replicating themselves. And these are present in the blood, which can be separated also, but the amount of blood needed for the isolation of the interferons is very enormous. So it is not practically applicable. So we cannot do it because large or enormous quantity of blood is required for isolating interferons from the blood. So to overcome this issue, interferons can be produced by rDNA technology. Already we discussed rDNA technology. It is used in the production of human insulin called humulin and also human alpha lactalbumin, which is a special protein present in human milk. So already rDNA technology is used to produce these two human insulin, that is humulin, and also this lactalbumin. Apart from it, rDNA technology can also be used to synthesize interferons. And for this purpose, the yeast called Saccharomyces cerevisiae, it is chosen. It is more suitable for the production of recombinant interferons compared to E. coli, because E. coli does not have the machinery for glycosylation of proteins. E. coli does not have the machinery for glycosylation. Glycosylation, it refers to, it's a particular step which makes the proteins to be stable. So proteins become stable by this step called glycosylation. So for this particular step to occur, Saccharomyces cerevisiae is supporting it, and because it has the machinery, but the E. coli does not have the machinery for the glycosylation of the proteins. And interferons are used for the treatment of diseases like cancer, AIDS, multiple sclerosis, hepatitis C, and herpes zoster. So these are the diseases which can be treated using interferons. But the problem is, though it has lot of therapeutic applications, that is to treat the patients. Interferons cannot be produced because not that much easily, because it is so costly. The high cost of the production of interferons is still affecting people, that is common people. So it is out of their reach. So common people cannot even think of going in for this treatment by interferons, because the cost of interferons production is so high that it's not reachable for common people. Otherwise, interferons, once used for the treatment of these diseases, people can recover themselves. So interferons are the special type of proteins, and they are antiviral in nature and species specific, and they are produced in the body cells itself whenever body cells are infected with virus. So cellular DNA is stimulated by the interferons to synthesize interferons. Cellular DNA is stimulated by the interferons to synthesize enzymes which act against viruses, and also viruses cannot replicate themselves. So interferons stimulate cellular DNA or DNA present in the cell, so that enzymes can be synthesized which stop viruses from replicating themselves, so that cells can be protected. Though interferons are so much useful in the treatment of diseases, due to its high cost of production, it is not within the reach of the common people. And this interferon can be easily isolated from blood, but it requires lot of or enormous quantity of blood, so it is not practical. So this rDNA technology is useful in producing interferons. And for this purpose, yeast Saccharomyces cerevisiae is more preferred than E. coli, because the Saccharomyces cerevisiae, it has the mechanism for glycosylation of proteins to make the protein stable, structure of the protein stable. So this step can occur only by this yeast Saccharomyces cerevisiae, because it has got the mechanism and not the E. coli.